So today we're talking about two things. The first one is the nephron structure, and the second one is ultrafiltration. So this is a part of the chapter called homeostasis, and to be specific, I'm looking at the osmoregulation, but specifically on the kidney part. So so here I'll be talking about the nephron structure first. So if you don't know what a nephron is basically like a structure like this. Um, well, it's like tiny functional structures inside a kidney, and it kind of spans from cortex to medulla. It's like a long tube, and okay, this is how I usually think of it. So this is like a simplified diagram of it. Um, I treat one nephron as having two components. One of the components being the kidney tube. By the kidney tube, I'm referring to like the kidney. So like you see like this part, this entire like winding thing. This is part of the kidney. And the other part I consider is the bloodstream tube. By this, I'm just talking about like the blood capillaries, the blood vessels that are like twirling around right here. But anyways, as you'll see later on, being able to differentiate between the kidney and the bloodstream will be really useful for you to understand selective reabsorption. But today I'm not going to talk about that. I'm going to talk about nephrons first. So essentially what we have to know about nephron is that there are several structures that we have to know about. So we have a cluster of blood capillaries called glomerulus. We have a Bowman's capsule, so this is already part of the kidney. And we have proximal convoluted tubule, that's like the beginning of the tube, like the front part. And then we have the loop of Henle, which is like the thing down here. We also have the distal convoluted tubule, which comes after the loop of Henle. It comes after that. Finally, we have the collecting duct. So that's the structure. I don't have another labeled version, but this is roughly the idea. If I was to like kind of label this thing, I'm gonna erase it later. But this is the Bowman's capsule. Inside here we got like a ball of capillaries. This is called glomerulus. We have this top part being the PCT, proximal convoluted tubule. We have this entire thing here being the loop of Henle. And I believe this is the descending part. This is the ascending part. So here you go. And this part will be the DCT, distal convoluted tubule. And this will be the collecting duct. And that's it. And just wanted you to know, this entire top bit is like the cortex. It shows up in the cortex of the kidney, while this part, basically the bottom half of the collecting duct and the bottom half of the loop of Henle, that's found in the medulla. So just to let you know. Okay, so now we're done with the we're now done with the nephron structure. So now I'm going to um proceed to the formation of urine. Okay, just before that though, I want to expand just a little bit more on the kidney tube and the bloodstream tube, like the two ideas I talked about, these two. So by the kidney tube, essentially I'm referring to the nephron, the entire nephron, so like the Bowman's capsule, the PCT, the loop of Henle, the DCT, and the collecting duct. So whatever that belongs to the kidney, I call it the kidney tube. And for the bloodstream tube, I'm referring to all the blood vessels that are involved. So that includes the renal artery, so from the bloodstream to the kidney, and the blood comes from the heart. Right now it's unfiltered. Unfiltered meaning we have everything, including urea, uric acid, and creatinine. So all that we don't want, but it's still there, unfiltered. I can also call it dirty blood. So the dirty blood came from the heart through renal artery. Now it reaches the kidney. And from renal artery, the blood enters afferent arteriole. So be careful, it's afferent with an A. So the afferent arteriole then leads to the glomerulus, which is a ball-like cluster of blood capillaries. And it's then after the glomerulus, it leads to the efferent arteriole. And then after the efferent arteriole, the blood goes to the capillaries. The capillaries meaning like the capillaries, like these things are like surrounding the kidney. Sorry, I was blocking it. And then after all the filtration have taken place, the blood is now cleaned. It's going to go back to the renal vein. So from kidney, it's going back to the heart. And right now, the blood that leaves the kidney will be filtered. I can call it clean blood if I want to. So that's all I want to say. So now I'm going to enter the first stage of urine formation. So it's called ultrafiltration, and we know that it takes place in the Bowman's capsule I have right here. And by the way, this is like a gigantic diagram that I found on the internet. So yeah, the basic overview of ultrafiltration is that it is the formation of glomerular filtrate in the Bowman's. Wait, let me rephrase that. It is the formation of glomerular filtrate in the Bowman's capsule. Yeah, that's it. So where does it take place? So for the kidney tube that are involved, that is the Bowman's capsule. That is this yellow thing. This is a Bowman's capsule. And for the bloodstream tubes that are involved, so here I have the afferent arteriole going in. Inside here, I actually have like a cluster of capillaries called the glomerulus. So that is also a bloodstream tube. And then the blood exits through here. This is called the efferent arteriole. So afferent arteriole, glomerulus, efferent arteriole. These are the bloodstream tubes are involved. Okay, let's look at a more straightforward diagram. So I actually want to show you like a website. This is a 3D model I'm looking at. Definitely not mine, but it's quite useful, and so I'm borrowing it. So this is a 3D structure of a glomerulus. So you can see like this part, there's the red bits. These are the what I call bloodstream tubes. So you can see we got like the afferent arteriole down here. And then I have this entire ball of glomerulus. And then now I have like efferent arteriole going out. And if you see, this entire ball of capillary is actually wrapped inside a kidney structure called the Bowman's capsule. And this Bowman's capsule actually has like a few adaptations that I'll talk about later. But you can see right now, it's kind of like a balloon that's covering the entire glomerulus. And there's a lot of free spaces as well if you look closely. There's like a lot of gaps, and that's actually really useful for our discussion later as well. So now we're done with the model, let's go back to the notes. Okay, so I'm gonna talk about the structure of Bowman's capsule. So from here we'll get like a little bit of the idea of what really happens during the ultrafiltration. So I think the first thing is to talk about if you look at the diagram here, I mean, it's not a really straightforward diagram, but essentially what's going on in ultrafiltration is that we want several things to stay in the blood because we can't risk it to be in the part of our urine. So ultrafiltration, it allows like the soluble substances to like enter the kidney tube. The soluble substances, there's actually a lot. I'm gonna name them. So definitely water, and in fact I think water is where everything is dissolved into. We have water, we have glucose, we have amino acids, we have vitamins, we have inorganic ions. So a few examples will be, I think, sodium. Well, just let me check. Yeah, sodium, potassium, chloride ion. So these are the things that are soluble, and these will become part of the glomerular filtrate. Okay, sorry for the long time to work out like how the pronunciation works. Okay, there's also several things that will be contained in the glomerulus filtrate. These are urea, uric acid, and creatinine. These are actually like the unwanted substances, and as you go on and learn about the selective filtration, you will know that these are actually not getting excreted through the urea. These are actually being excreted through the urine. Sorry, I keep saying the wrong thing. So these are being excreted via urine. I think the way I'm talking today. I'm also gonna confuse myself later, but that's because I'm a bit like dizzy right now, so I'm sorry about that. Okay. So essentially what the ultrafiltration does is that we don't want certain substances to pass to like enter the kidney. So certain things are blocked in ultrafiltration itself. These are red blood cells, white blood cells, and large plasma proteins. So essentially, anything that's larger than a relative molecular mass of 69K will be blocked. Okay, that's pretty much it. I'm gonna write on top. Wait, this is called ultrafiltration. So now, in order to let the soluble substances, i.e. the fluids, pass, and blocking the large stuff, surely there are some like adaptations both for the kidney and for the blood vessels so that this can happen, right? So there are some like adaptations. So yeah, there's actually like quite a lot of adaptations. I'll go through them one by one. Okay, the first thing we're gonna look at is the endothelium of the glomerulus. Let me just quickly find it. The endothelium, okay, wait, let me paste a picture from the model we just saw just now. That could be useful, maybe, I'm not sure. Okay, so if you now look at this blood vessel, right? Like the blood vessel, the afferent arteriole, this glomerulus, then the efferent arteriole. If you just look at this part, so you know that blood capillaries, right? They always have like capillaries that kind of, I think it will kind of look like this. Like, you know, blood capillaries, right? They're actually not like back to back. They have these like little gaps in between, and that actually allows like the interaction between tissue fluids and blood plasma. I'm not going to talk about it here, but just know that the capillary wall is actually having like a lot of large gaps. So that's kind of how like the blood capillary usually looks like. And just to let you know, this structure is part of the glomerulus. And they have these like gaps or fenestrations. Now this acts as the first filter, and essentially the endothelium of blood capillaries, the most that they can do is to filter out some of the red blood cells, some of the white blood cells, but not everything, as they're not like a perfect filter. So I'd say most of the stuff still passes out, like all the water, glucose, amino acids, ions, vitamins, urea, uric acid, and creatinine. They still pass they still pass out of the endothelium, but certain things like the red blood cell, white blood cell, and large plasma proteins, some of them are blocked and some of them are not. So if we only have this one as a barrier, and what if some red blood cells pass through the filter and enters the kidney? That's not something that we want, because we don't want to have blood in our piss, right? So now we have a second structure called the basement membrane. This basement membrane is on top of the endothelium of the glomerulus. This basement membrane, it's formed by collagen and glycoproteins, and it actually acts like a mesh. So you can imagine like a mesh, a mesh would be something like this, really tightly packed, nothing really can pass through. And this is actually the main selective barrier. So this basement membrane, this is actually the main filter for the Bowman's capsule, because this prevents all of the red blood cells, white blood cells, and large plasma proteins from passing through the bloodstream into the kidney. So yeah, this is the main thing. And remember, as I've said just now, any substances with a relative molecular mass that's larger than 69,000 cannot pass through. So that's the basement membrane. There's actually something else beyond the basement membrane, and however, I want you to keep in mind, because now we've essentially we've done the process of filtering out any blood cells and large proteins. Now the function of the third structure is a little bit different. It's not really about filtering anymore, and I'll talk more that a little bit more later on. The third structure is called epithelial cells of the Bowman's capsule. So keep in mind, this actually comes from the Bowman's capsule. I'll try to demonstrate it here by extending it a little bit, probably. I'll actually draw this using a highlighter. You'll understand my drawing later. So this extends from the Bowman's capsule, and it acts like I like to think of this cell as a octopus, as it kind of just wraps around the entire glomerulus, actually in the entire thing. It just wraps around it like this. So you might be wondering, so what the hell is going on? So this is actually the epithelial cells of the Bowman's capsule. We also call it podocytes. And it is, in fact, the inner lining of Bowman's capsule. And just to keep in mind, this is part of the Bowman's capsule. It is part of the kidney. So it wraps around the capillaries of the glomerulus, and these are actually, maybe I didn't draw it well, but these are actually finger-like projections that can form gaps and filtration slits. So that's what they say by finger-like projections. And by filtration slits, maybe you can refer to this picture. You see do you see the gap like here, here, here? These are gaps, right? I'd say these are like pretty large gaps. And these are what we call as the filtration slits. Now you might be wondering, but we already have filtered out all the big substances, so why do we still need this one? And technically if it has like a great gap, it's not gonna filter anything. It's kind of useless, right? Well, no, this is not useless. It's not useless at all. And I'm gonna explain why. So, have you ever thought about, for ultrafiltration, we want ultrafiltration to be occurring at a higher rate as well. The higher the rate is better. So, the glomerular filtration rate is actually really fast. So meaning this entire filtering is actually, it's actually quite fast. So what is it really fast? There's actually two factors playing a role here. And well, I think surprisingly, out of the two factors, only one of them enables the filtration to be faster. One of them actually slows down the filtration, but like the overall effect, I think the factor that enables the process to be more faster actually wins. Okay, so which are the two factors, you may ask? So first thing I want to say is that the glomerular filtration rate is actually high. You may ask why. The two factors playing a role. So the first one would be the water potential inside bloodstream is larger than the water potential in kidney 2. Okay, so what do I mean here? There are actually several structures in play that enables like a steep water potential gradient between the bloodstream and the kidney. There are several structures, so one of them being the afferent arteriole has a larger diameter than the efferent arteriole. If you look at the arterioles, they didn't label them, but let me go to a better diagram. Essentially, they're saying that this arteriole has like a larger diameter than this one. So remember that the afferent arteriole actually delivers the unfiltered blood, from the heart into the glomerulus. And in the glomerulus is what the ultrafiltration happens. And the ultrafiltration, in order for it to be carrying at a high speed, because remember, this is like the direction of water flow. So essentially, if you want water to flow out fast, you better have like a really high hydrostatic pressure inside this glomerulus. That's like the way if you want water to exit the glomerulus fast. So one way to do that is we have a much larger afferent arteriole than the efferent arteriole. So you can imagine wait, I keep writing the wrong thing. This is afferent arteriole. This is efferent arteriole. You can imagine that as the diameter decreases, right, the hydrostatic pressure, not here. So when the diameter decrease, the hydrostatic pressure, oh my god, increase. We could also say that there is a much higher water potential inside the glomerulus than the Bowman's capsule. We can also say that. And essentially the result of that is that the water is going to like flow out a lot faster than if there's not this difference in diameter. And that's only one of the structures. The second structure is the podocytes that are wrapping around the glomerulus. So this podocyte, just now I thought would be useless, but this actually provides a pressure. So this entire wrap thing is giving like a pressure to the glomerulus. And this pressure added can also it can also like squeeze out the water. If you imagine like a person is squeezing a balloon, it can squeeze out like the water a little bit, and that is actually part of the podocyte's function. But however, you have like a problem here. So even though I said the water will be flowing out a lot because of the difference in water potential, also keep in mind that inside the glomerulus, we have a lot of we have red blood cells, we have white blood cells. Wait. Imagine that's like white blood cells. Okay. And we also have like large proteins. Wait, same color. Large proteins, maybe like this. This means that we have like a lot of solutes. Well, actually much more solutes, large solutes, than the Bowman's capsule. So here we actually have more solutes. And here we have less solutes. So water also tends to flow from a region where there's like have a less solute concentration to a region of higher solute concentration. This is also a tendency of water flow right here. So this actually counteracts with what we were achieving just now. And this, eventually, it actually slows down the glomerular filtration rate. So this is the second factor. The first factor being the one I just talked about. It's the difference in diameter, and also the podocyte structure wrapping around, giving giving some hydrostatic pressure that forcing the water out. And that's like the first factor. And it increases the glomerular filtration rate. So if you ask me, so now I have two factors. One increases the rate, one decreases the rate. So definitely one of them wins, because we definitely will have to have water either flowing in or out, not in between. So I'm going to tell you the factor that wins is the first factor. As there is much more water flowing out of the glomerulus due to the two adaptations I've talked about, than the water that flowed in because of the higher concentration of solute in the glomerulus. So this factor 1 actually outweighs factor 2. And I hope this helped. I think this is all I want to talk about. And just before I end, I want to make it clear. So now, after the ultrafiltration, what do we have now? So we have successfully produced the glomerular, wait, not actual ruler, glomerular filtrate, not filtration, oh my god. Okay, this is not working. The glomerular filtrate now contains all the soluble substances, so like water, glucose, amino acids, vitamins, mineral ions, and also like the less useful one, like urea, uric acid, and creatinine. So what's not in the glomerular filtrate are the red blood cells, the white blood cells, the plasma proteins, and also anything that is larger than 69 K relative molecular mass. And that's it. Thank you for listening, or not.